Okay, welcome to part two on our series on cell transport. And in this section of notes, we're going to talk about tonicity, um, predicting what way materials are going to flow in different situations involving cell membranes, and a little bit on filtration right at the end. So let's go ahead and get started. So, the effects of solutions on varying tonicity. So, there's basically three different states you can have when you're talking about the concentration of water and solutes inside a cell and outside of a cell, and trying to predict which way those water and solutes are going to want to flow, assuming the cell membrane will let them flow. There are three situations. One is isotonic, that we call hypertonic, and the last we call hypotonic. Okay. Isotonic, the top one, iso means same. So this is a situation where we have the same concentration of solutes inside and outside the cell. And let me just break away for a second. The tonicity refers to the level of solutes, and we're always looking, the term always applies to the level of solutes outside the cell. So in the fluids surrounding any particular cell, we describe that fluid as being isotonic, hypertonic, or hypotonic based on whether it's got more solute or less solute than what's inside the cell. So isotonic means we have iso same amount of solute outside the cell as we do inside the cell. So essentially, we're already at equilibrium. This is everything's kosher, everything's copacetic. If things can pass through the membrane, they're passing back and forth, but they're passing back and forth at roughly the same rate. There's no big downhill trend moving things in or out. Everything just stays calm and peaceful and just where it's at, and everybody's happy. Now, on the other hand, hypertonic, hyper means lots, right, or more. Like if you got a hyperactive kid, it's somebody who's really active. So hypertonic means really tonic, or lots of solute, or greater amounts of solute, or concentration of stuff outside the cell than inside the cell. Okay, so in that situation, so we got lots of solute outside, very little inside, and that gives us a low water concentration outside, right? Because we got lots of solute, it's kind of like seawater, right? Lots of solute means less water. So outside we got high solute and low water in hypertonic solution. So in that situation, and we'll do some images and stuff on the whiteboard in just a minute to help clarify all this. But water is going to move out of the cell, right? Because we got a low concentration outside. Water goes out. And solutes, if they can pass through the membrane, will go in because high outside, low inside. So hypertonic is hyper or high solute outside the cell. Water will tend to move out, and solutes will tend to move in, assuming they can pass. And the last of the three possible situations is hypotonic. Hypo means low or beneath. Think of a hypodermic needle when you get a shot. Hypodermic means below the dermis or below the skin, that second layer of skin. We'll get into that when we get into the integumentary system. But it's a needle that's meant to go below the skin. So hypo means below. So hypotonic means less solute. Okay, less solute outside the cell than in, which means you have a higher water concentration outside. So less solute outside means you're closer to pure water, right? So pure water would be a super hypotonic situation. So high water outside, water is going to want to move in, like we talked about with the plants in the last section of the notes. Water moves in, adds pressure inside the cell, and solutes may move out if they're able to. Okay. So let's take a look here. I should probably get myself out of the way. So here are some examples of what cells might look like if you put them in the different solutions. So if you put a cell in a hypotonic solution, hypo, low solute outside the cell. So if it's low solute out here, that means it's high water. So water is going to want to try to get in. And sometimes it can be so much that it'll actually burst the cell. We call it lysing it. And here's a look at a plant cell down here. It's turgid, right? We fill it up. It's like putting your tire under high pressure. It's nice and firm. This is that celery sitting in the water situation. Isotonic, equal concentrations of solute and water on both sides. Water flows in and out at roughly the same rate, just kind of at a normal cell turgidity, okay? Maybe slightly flaccid if it's a plant. And hypertonic, meaning hyper, lots of solute outside the cell. So lots of solute means low water, kind of like salt water, right? It's going to want to flow out, and it's going to shrink your cells, shrivel them up, or plasmolyze is what they call it with a plant cell. All right, so let's go to the whiteboard and go through a few more of these. All right, so we're back from the whiteboard now, and just a couple animations to hopefully help show you a little bit of what I mean, maybe with some better animations of what I can draw. So let's take a look at some examples of hyper, hypo, and isotonic situations that I'll draw out for you here. So I'm just going to draw kind of the classic example. I'll just draw a beaker, and we'll put a cell in there. I should do a cell in black for the cell membrane. So there's a cell, and it's sitting in water, and we'll leave the white space will be the liquid water, and I'll make the solute, let's call it salt, with the little green here. So if I do, all right, what would you call that? Yeah, I'd call it isotonic. We have roughly equal concentrations of solute outside the cell as we do outside the cell as we do inside the cell. So we'd call that isotonic. Okay, so it could be, let's say, 20% solute inside, and it would be 20% solute outside. Now, if that's the case, what would be the water percentage here? Well, if it's 20% solute, it's 80% water inside and 80% water outside. Let's do the same thing, but change up the percentages a bit. Okay, so we will draw our cell, and then we'll put in our solute. What would you call that situation? Yep, that would definitely be hypo. Hypo, low solute. And remember, it's solute outside the cell. Okay, so in this situation, the solute might be 80% solute inside and maybe 20% outside. And that would put the water concentration at 20% inside and 80% outside. So now let's figure out which way, if water can pass through this membrane, which way will the water go? Into the cell or out of the cell? Right, it's going to go from high, 80, to low, 20, out of the cell. Now, if solute can pass through in this hypotonic situation, which way will the solute pass? Yes, it'll pass out, high to low, big to little, again. If it can pass through. If it can't pass through, it's going to bounce off. It may want to go out, but it can't. It's going to get stuck inside that situation. And we'll take a look at that in some animations in a minute. Let's reset and try this again. Okay, so we'll draw our cell, and we'll draw our solute. What would you call that situation? Yep, that is definitely hypertonic. Hyper meaning more solute outside. So let's just make up some numbers here again. Let's say our solute outside is, let's say 75% solute outside and 15% inside. Well, that would mean, what would the percentage of water be inside the cell in this situation? Yep, 85%. And what would it be outside? 25%. If water can pass, which way will water go? High to low. It'll head out. And solute, high to low, it will go in. All right, assuming it can pass. If it can't pass, it won't, and we'll just have the water moving, and our cell will shrivel all up because it'll lose all the water, but not gain anything else. And so anytime you're asked to predict what way things are going to go, and we'll ask that specifically on the test, and we'll also ask it for different situations in the muscle and nervous and digestive and respiratory systems and the cardiovascular systems are all going to deal with this type of situation. You can just draw a picture like this. Okay, draw out the concentration inside the cell and outside the cell, and you just draw your arrow from high to low, from the big number to the small number. Okay, and then if it can cross the membrane or not. That's all you got to keep in mind. Things are going to move from high to low, big to small, if they can pass through the membrane. And the tonicity, hyper means more, and it's always referring to the solute outside the cell. Hypo is the opposite, means less solute outside the cell. And let's take a look at a couple animations and see if that helps as well. So here we have a cell membrane, phospholipid bilayer, a couple of protein channels here. And what we got going on, move myself all the way a little bit. What we got on here, we've got lots of water inside and very little solute, lots of solute outside and very little water. So this is a hypertonic solution. Okay, hypertonic. So water is it's able to pass through here in this situation. It's going from high to low, and you can see how it's shrinking the cell a little bit because it's trying to reach equilibrium. So we got roughly equal concentrations of water and salt on both sides, but that means less water on the inside because the solute couldn't pass through. All right, so here's another animation. We can see a hypotonic solution on here. We're in the cell, out of the cell. Blue equals water, and the green things are the solute. So we got very little solute, low solute compared to the inside, which means we got a lot higher water concentration out here. Here we have isotonic, relatively equal on both sides, and here we have hypertonic, hyper, lots more solute outside the cell than inside the cell. Okay. So here we're going to see an isotonic solution. We'll hit play. And we can see things moving in and out just fine. What I like about this is it does show that the water does need some protein channels to go through, which our last one did not. All right, so we got relatively equal movement. Nothing is really changing. Okay. Here, what situation do we have right here? Take a minute and tell me whether this is hypertonic or hypotonic. Yes, we have less solute outside, so low solute outside compared to inside. So this is low or hypotonic. And now take a second and tell me which way water and solute is going to move if it can pass. Let's see if you're right. So if you said water was going to move in, you are correct. In a hypotonic situation, water will move in. And if the solute could have gotten out, it would have, but in this situation, it wasn't able to pass through. So water moves in a hypo, low solute concentration situation. Let's take a look at the next one. So take a look at this one and tell me whether you think this is hyper, hypo, or isotonic. All right. Let's see if you were right. All right, so if you said this is hypertonic, you are correct. We have a lot more solute outside the cell, a lot more of the green solute outside the cell, less inside. So in a hypertonic, we've got a higher water concentration inside the cell, so water is going to want to move out. And we will see that happen in just a second. And the cell shrinks. All right, let's go back to the notes then. So our last slide on this section of notes is transport called filtration. Now it's as far as the membrane's not doing any actual work here, so it's passive, but there is energy being expended by the organism. So not necessarily the cell that the material is passing through, but by the organism. And this is called filtration. This is the passage of water and solutes, depending on how your membrane's set up, powered by hydrostatic pressure. So just water pressure. If you squeeze a balloon, you can make more water come out of it faster because you're increasing the pressure. Okay. So the pressure gradient, not concentration gradient, not high to low concentrations, but high to low pressure, will push the fluid across from high pressure to low. This is what happens in our kidneys. We filter our blood in our kidneys. So the blood in the kidneys is under higher physical pressure than the areas that collect the urine. So it will force material from the blood into that urine collection area where we can then filter it out. Okay, so that's filtration. It's membrane passive, but it is an active activity and it requires a whole organism and tissues and organs working together as opposed to something that's happening solely at the cellular level. All right, and that will do it for us on part 2 of the no-sun cell transport. In the next part, we will talk about active transport, where cells actually use their own ATP energy to move things around. So we'll see you next time.